procedure. The new age of biomedical engineering, CRISPR gene editing. Having a relative or a close friend who has a genetic disability or Alzheimer's due to age, CRISPR-Cas9 gene editing engineering may be able to revolutionize our world with these diseases. So what is CRISPR and how did it come to be known to mankind? What are the potential uses of the CRISPR system and the CRISPR's clinical trial of 2022? So first, let's examine what is CRISPR gene editing system. Well, according to MD Anderson Cancer Center, CRISPR is short for clustered regularly interspaced short palindromic repeats. It enables precise DNA editing by utilizing a CRISPR-associated enzyme such as the Cas9 protein, making a molecular scissor that targets specific DNA locations. This technology allows scientists to edit the genome of living organisms by introducing new DNA fragments, allowing disease-causing mutations to be replaced with healthy sequences or other genetic modifications. Additionally, CRISPR facilitates gene knockout, a common method for studying the gene functions and allowing for modifications in gene expressions known as epigenetic editing. These advancements provide scientists with potent tools for disease treatment, agricultural advancement, and exploration of fundamental biological inquiries. CRISPR's history and background, initially identified as E. coli bacteria DNA sequences by Ishino at Osaka University in 1987, CRISPR, or clustered regular interspaced short palindromic repeats, presented a challenge in sequencing due to its complexity. However, with the groundbreaking demonstration of CRISPR-Cas9's potential in human cells, discussions on safety, ethics, and the potential for gene therapy intensified. Notably, Jennifer Doudna played a pivotal role by organizing a conference in Napa Valley, California in 2015, which delved into the scientific, medical, legal, and ethical dimension of genomic modifications. And next, I will tell you CRISPR's potential uses. According to National Center for Biological Information and National Library of Medicine, CRISPR-Cas9 offers exciting potentials for treating genetic disorders stemming from single-cell gene mutation, and cystic fibrosis, Duchenne muscular dystrophy, and other genetic disorders. Furthermore, it holds promise for combating infectious diseases such as HIV. Despite the effectiveness of current HIV therapies like antiretroviral drugs, a definite cure remains elusive due to the virus's integration into the host's genome. Research has showcased the ability of CRISPR-Cas9 system to target HIV-1 genome activity, effectively suppressing gene expression and replication in various cell types by harboring latent HIV infections, all without inducing toxicity. Additionally, this method shows potential for immunizing cells against HIV-1 infection, and there is a growing interest in leveraging CRISPR-Cas9 to modify patient-derived T cells and stem cells, which could then be reintroduced to patients for disease. Finally, CRISPR's clinical trial in 2022, the inaugural CRISPR-based trial for sickle cell disease and transfusion-dependent beta thalassemia, was jointly sponsored by Vertex Pharmaceuticals and CRISPR Therapeutics. Their methodology involves utilizing CRISPR to re-enable the expression of fetal hemoglobin in cells. Currently in phase 3 trials, this treatment is under evaluation in both adult and pediatric populations with severe SCD or TDT. Preliminary results from over 40 individuals with TDT and more than 30 with SCD have been reported, demonstrating remarkable and sustained efficiency. Out of 44 participants with TDT, 42 were no longer dependent on transfusions following treatment, with follow-up extending up to 3 years. Similarly, all individuals with SCD remain free from vaso-occlusive characteristics of the diseases after treatment, despite not directly correcting the underlying mutations responsible for SCD and TDT. Their approach appears to offer functional cure for both conditions. There are currently many clinical trials for CRISPR to treat various genetic disorders such as blood disorders, diabetes, cardiovascular diseases, immune disorders, and many other disorders. In summary, CRISPR is a powerful gene editing technology that originates from bacterial defense systems against viruses. Discovered in the early 2010, its development revolutionized genetic engineering due to its precision and accessibility. Despite its potential for curing disease, enhancing crops, and advancing technology, CRISPR raises ethical concerns regarding its implications in human germline editing and environmental impact. Clinical trials employing CRISPR have shown promise with ongoing research focusing on treating genetic disorders, cancer, and infectious diseases. However, rigorous ethical oversight is essential to navigate the ethical complexities and ensure responsible use of this transformative technology. CRISPR will be the technology that our generation remembers and honored for the major biomedical breakthrough of CRISPR technology. Thank you.